A bacterial cell. The workings of this seemingly simple unicellular being has intrigued scientists for centuries. Bacteria have been on this planet way before us and have perfected the art of survival. They are complicated, simple beings, a paradox of sorts. Let's dive in to see what goes on inside the cell. In this case, we have a model gram-positive bacterium. Once you peel off the thick peptidoglycan wall and the plasma membrane, you reach the cytoplasm where all the action takes place. Here you can see bacterial DNA being transcribed to mRNA, which is then translated to protein. Bacterial DNA is compact, and this relatively tiny piece of nucleic acid codes for thousands of proteins. Some of these proteins being popped out of the ribosomes have to find their way to the destination on the membrane while navigating through the bacterial milieu. Organized chaos. The cell division machinery are the most well-studied group of proteins. Its players interact with each other directly through intermediate partners as well as sensing membrane curvature or specific lipids. Nearby, we have the co-localized secretion and sorting machinery. Multiple proteins focalize at the membrane to form complex machineries. If we move further from the septum, we can see that conjugation machinery is temporarily at the cell pole, but may disassemble when the cells enter different stages of growth. Assembly and disassembly of proteins during the different cell stages of growth is a fascinating process. What triggers disassembly? Is it always de novo reassembly and disassembly? Or is there migration of these proteins from one spot to another? Even though we know quite a bit about interaction and localization of the main players of different cellular machineries, the whole hierarchy is far from clear. But why is this hierarchy so important? Imagine a focalized cellular machinery as a game of Jenga. Each Jenga block symbolizes a protein in the machinery. Proteins assemble in a certain order, and studying them within a network of interacting partners helps us see the bigger picture. If we only look at, for example, this protein here at the top, we do not necessarily understand the entire picture, or for that matter, the impact this protein has on the machinery and vice versa, just like in this Jenga tower. If I were to remove the top block, nothing really happens. It may be a little less stable, but still functional. While if we only study the base proteins, or the one in the center, we disrupt the entire process and miss out on understanding what these other proteins do in the system. If I were to move the base block, the entire tower would come crashing down. Focus has now started to shift to studying these proteins in networks rather than as solitary players. Developing imaging tools will rapidly advance our understanding of protein dynamics in prokaryotic cells.